So this is the nervous system lecture number 2 that goes along with the Hole's Essentials of Anatomy and Physiology text. And lecture number 2 is regarding the synapse. So when you have two neurons, they don't actually touch one another. They come really close to one another, but there is a little gap between them. And that gap, that junction, basically, it's called a synapse. And it can be between two neurons, or it can be between a neuron and a muscle fiber, like we learned in the last chapter. So it's very, like they come very close, don't actually touch. If they actually touched, we wouldn't be able to control the neurons. They would always be firing, which would be kind of like if you've ever been shocked, had an electrical shock, how it's always being stimulated. So there's a gap there so that we can basically turn it off. We don't need our synapses firing at all times. The gap itself is called the synaptic cleft. And the synapse is made of the two neurons and the synaptic cleft in between them. So the neuron that sends the signal, which is called the impulse, that's presynaptic, and the one that receives the impulse is postsynaptic. So at least that part makes sense. Now they do not touch, so they don't communicate through an action potential like we're going to learn about. Instead, one neuron releases a neurotransmitter, and then that hits the neurolemma of the other neuron, and that triggers, a chemical reaction within the postsynaptic neuron. There are many different kinds of neurotransmitters. You may have heard of a few. There's, we've already talked about acetylcholine, but there's also dopamine and serotonin, GABA, cannabinoids, melatonin, and others. They're very complicated, and we're still kind of learning what they mean. But it's basically like how humans use words to talk to one another. Neurons use these chemicals. They basically like throw chemicals at one another to send a message. And decoding that message is rather difficult, especially since you can't just dissect a living brain and see what's going on as you ask that person questions. That would be unethical. So we're still learning. But the neurotransmitters are stored up in vesicles inside the presynaptic neuron, and when an action potential has traveled down that axon, that stimulates those vesicles to release the neurotransmitters through exocytosis into the synaptic cleft. They'll diffuse across the cleft and then bind to receptors. So some people naturally learn certain things faster than others. So what that means is their neurons are a little bit closer together. So the distance on the synaptic cleft is shorter. And it's different for different people. Some people, say pick up music really quickly, and other people would pick up sports really quickly, or reading. It just depends. But you can, like the process of learning is actually growing your pre and post synaptic neurons closer together along the pathway that's used in whatever skill it is you're learning. So I'm sure you've heard the phrase, if you don't use it, you lose it. Same thing if you're not using a skill, like speaking another language, over time your body will actually recede. Those neurons will kind of get further away from each other because it takes energy to keep those neurons close together. The cytoskeleton has to grow and push the axon into that configuration. So learning something takes energy. It's also why students are often tired at the end of the school day. Besides from getting up so early, that you have to grow those neurons closer together. It takes ATP. Your brain consumes an enormous amount of energy compared to the rest of your body, like a quarter of all your calories. But over time, as you learn something and as you practice and reinforce that skill, you can recall and retrieve that information faster, which is why, especially for sports or a musical instrument, you have to practice to get that muscle memory. Here's a picture of a synapse. So here's an axon, and the dendrites receive the impulse and they send it to the axon hillock and then down the axon. So there's a one-way direction of travel. So this neuron is making a synapse with this neuron. Here's an axon here, and the impulse is going this way. Here's a up close view of the synaptic cleft here. So this is the knob where the neurotransmitters are stored, and there's a teeny tiny gap, the synaptic cleft. And here's a closer up picture. If you look, there's a presynaptic and postsynaptic neurons. So these are the neurotransmitters. They go through exocytosis, the vesicles merge with the cell membrane, release the neurotransmitters, then these are receptor proteins on the cell membrane of the postsynaptic neuron. And once enough of these are triggered, that will start a new action potential. There's a little mitochondria here too. This takes a lot of energy, so neurons have a lot of mitochondria in them as well, as just like muscle fibers do. This is a picture of an electron micrograph. So this is like the actual molecules, what they look like. The only thing that is different is we've added color to it to make it easier to see. But this is what they do look like in a cross section. So here's all the mitochondria. Notice they're circular, they don't look like an oval. That's because they're just the cross sections like on the other axis. But here's all the vesicles with the neurotransmitters and a teeny tiny synaptic cleft. And then here's the postsynaptic membrane. Okay. Now this is the more difficult part of this unit. So when a neuron is at rest, okay, it is polarized. So what that means is inside the cell and outside the cell, on either side of the cell membrane, they have different charges. And the brain, the body does this by moving ions back and forth, okay? So the charge is different. So there's a charge differential, which means it's polarized, kind of like a battery. A battery has a positive and a negative end. Same thing here. The ions are unequal, so that makes the inside of the membrane a little bit more negative than the outside of the membrane. So because there's a charge differential, when you open up any transport proteins, ions will move just naturally without any energy added by the cell to even out that charge. This polarization is how we can send nerve impulses, or how our neurons can send nerve impulses and make an action potential. So the action potential is involving a specific change in membrane polarization, and then eventually it will return back to the resting state. So this is a process, and it'll happen over and over and over again down the cell membrane all the way down to the end of the axon on the neuron. So there's a series of protein channels that we'll talk about that allow these ions to move, and the action potential travels down the axon, much like a wave, at a stadium, at a sporting event might travel with people standing up and sitting down. It's kind of the same thing. We call that propagation. The action potential propagates. And eventually, when it reaches the end of the axon, the end result is the neurotransmitter gets released into the synaptic cleft. And all this is done with ions. Okay, the two ions are sodium and potassium. Sodium is Na and potassium is K. They're unequally distributed. So when the neuron is at rest, so that's our starting point, there's a greater amount or concentration of sodium ions outside the cell. And then there's a greater concentration of potassium ions inside the cell. Okay, so that's important. So sodium's on the outside, potassium's on the inside. Okay. There are also many other ions involved in the cell. Most of them are negatively charged inside the cell. So that's part of where the negative charge comes from. Okay, and how those ions are arranged, whether they're on the inside or the outside, that's determined by different proteins that are on the cell membrane. They're called membrane channel proteins or pumps, depending on what they do. Each one is selective. That means it only lets a particular one in or out. So there's a sodium ion channel that only lets sodium in. There's a potassium ion channel that only lets potassium in. there's also a sodium-potassium pump that puts the sodium and potassium in a particular place, but you need both to make the pump work. So inside starts as a more negative, and outside the cell starts as a more positive. Okay, and potassium ions, it's super easy to get them to go through the cell membrane compared to the sodium. Okay. Now, a lot of these ion channels do have gates. So even though they are passive transport, they stay shut until something happens to open those gates. Kind of like at school, students stay in the classrooms until something happens. The bell rings and everybody, leaves to go to lunch or to the school buses. Same thing. Okay, so here's an example. So this would be an ion channel, but it's closed, it has a gate. And then this would be when it's open. So these proteins, they can change shape and keep control of what enters and exits the cell. Okay. So in the beginning, when a neuron is at rest, its charge is down here at the bottom. This is what we call its resting potential, but it actually has a charge. So if you measure like the difference across the membrane, and its charge is negative 70 millivolts. Okay, the M is for milli. So this is a tiny electrical charge, but we can measure it, and we do with EEGs, if you've ever seen on TV how they put those electrodes on someone's head, and then you get the little blips on a computer screen. That's what they're measuring. We could actually measure the electricity generated by our brains. So when these gates get opened, the sodium and potassium ions will rush in the case of sodium, or rush out in the case of potassium, through passive transport, because they're going from a higher to a lower concentration. You have to kind of think back to when you had freshman biology to remember that. So there's no energy necessary in the form of ATP by the cell. It just kind of uses what would naturally be occurring. So this, like I said earlier, the potassium travels through the membrane faster than the sodium. So potassium is quicker, and that causes the inside of the membrane to have the excess negative charge. Okay, now to reset a neuron to resting potential, that does require energy in the form of ATP. So the cell has to actively transport the sodium and potassium to where they need to be for resting potential. That active transport purposely puts all the sodiums outside and all the potassiums inside. So that's at the end. We want an action potential to happen quickly. So the resting potential is maintained at the end. So you have an action potential that happens, it doesn't need any ATP, and then at the end, the brain uses ATP, the neuron uses ATP to put everything back to restore that resting potential. It's kind of the equivalent of like setting a mousetrap. Okay. So the cell membrane is polarized. When an action potential does happen, it will, the first thing that happens is sodium will rush into the cell, and then potassium will rush out of the cell, and then the action potential is propagated. And then the third thing that happens is the sodium potassium pump puts the sodium back outside and potassium back inside to restore the resting potential. And then action potential is that like series of events. So sodium rushes in, potassium rushes out, and then the potassium pump puts things back how they were. This happens in a series, so there's not just one sodium channel and one potassium channel and one sodium potassium pump. There's a whole lot of them over and over and over again, all down the entire length of the axon. So the action potential happens in a wave. You can kind of see it here. they're showing you where the different charges are. This is in your book as well. Because neurons can do this electrical change-up, we say that neurons are excitable. Okay, sometimes we call it irritable, but they're not cranky. What it means is that they can respond to environmental and internal changes, and they can respond quite quickly. The response is changing their resting potential, starting an action potential, and then that triggers some action by the body over the long haul. Once the action potential is triggered, so you start at resting potential, then the sodium ions rush in, and we call this depolarization. So the inside was negative, but with all the sodium ions rushing in, they're positive, so that changes and raises the charge of the inside. So it's called depolarization. The stronger the stimulus, so like the more that neuron has other neurons sending it a message, the stronger the depolarization is going to be. So if once it hits a threshold level, so here it is, negative 55 millivolts, up from the negative 70, then the action potential starts. So you can see here, this is resting potential. This would not meet the threshold level in this case. On this axis is the voltage, the membrane potential, and over here is time. But once this crosses that threshold level, then action potential will occur. And you can see there's 3 presynaptic neurons here triggering this postsynaptic neuron to fire. So in that last picture, sorry, let me go back. You can see there's 3 neurons. Actually, only 2 are sending a message, okay? But down here at the axon hillock, these is where these charges will get summed up to determine if there's enough membrane potential. So this change in sodium will all kind of gather here. Okay, and that's also called the trigger zone. The axon hillock is the trigger zone. So once that threshold is met, the sodium ion channels open up and sodium ions rush into the cell, which changes the membrane potential, which causes depolarization. And then after that, but very close to it, the potassium channels will open, and that allows potassium ions to leave the cell. And then this is now called repolarization. So potassium ions and sodium ions both have a positive charge. It needs to repolarize so the action potential continues forward down the axon. If it stayed depolarized, then the action potential could kind of go back and forth, and there wouldn't be a direction, and that neuron would never pass the message on. So that's the purpose of the potassium ions rushing back in. And that reestablishes resting potential in that area on the axon temporarily. Sometimes it gets overly negative, and we'll call this hyperpolarization, hyper for too much. After that point, then the sodium potassium pump picks up and resets everything back to its original resting potential. And here's a graph kind of showing you. So this is the resting potential at negative 70 millivolts, and then a threshold level is reached here at about negative 55. Then you have a very rapid, you can see this is in milliseconds, so this is 1/1000th of a second. We could actually measure how long it takes a signal to travel through a neuron. Okay, so in less than half of a millisecond, you get this depolarization. And then another half of a millisecond, you get the repolarization, and then down into hyperpolarization until you get the resting potential being reestablished. And this is just putting the 2 together so you could see. Okay. And here's a graphic showing you how these events happen. So you can see there's potassium ions inside the cell. This is a close-up of an axon. That's why it looks like a tube. And then you have sodium ions outside the cell. And they're switching, the ions switch places, okay? That's where you get the depolarization first. The potassiums rush in, or excuse me, the sodium ions rush in, and then the potassium ions rush out, and then you get the repolarization. And this happens and it shifts itself down the axon, okay? And it happens like in waves. Okay, so this will happen and the impulse will be conducted all the way down the axon, this changing of the electrical current, okay? So you'll see, like here, you can see it a little bit better. So you have the action potential occurring here, and then the action potential occurs here, and then it occurs here, and it goes on and on and on and on, all the way down until it reaches the axon terminal. And here is a table, it's in your book, okay, of the sequence of events. Now, there is a difference between axons that have a myelin sheath and those that don't. The myelin sheath acts very much like the rubber coating on a wire. The myelin sheath insulates and helps to keep those ions close to the axon so they can't just diffuse away. So what that means is that the ions can move back and forth across the cell membrane really quickly because they're going to stay close by. The unmyelinated fibers, they conduct their impulses sequentially over the entire length of the membrane, which has the, that's really slow actually. And the example I just showed you in the picture was an unmyelinated fiber. It's kind of slow compared to the myelin sheath. Now with the myelin sheath, you have those nodes of Ranvier. If we're talking about PNS neurons, and what will actually happen is something called saltatory conduction. So you'll have a like an arc of the electrical signal passing from one node to the other as you move down the myelin sheath. So it kind of skips some steps. It's actually really cool, and it's super fast, which is great because let's say, for example, you you open the freezer and like a 5-pound pot roast that's frozen falls out and it's going towards your foot. Well, you need to pull your foot back, and the neuron that's controlling your foot goes all the way from your lumbar region in your back, lower back, down to your foot. That's really long for a neuron to get this action potential there. So because that's a PNS neuron, it has this saltatory conduction, so the electrical signal can kind of arc and skip forward and pull your foot back very quickly, assuming you're paying attention so that you don't smash your toes. So as you can see down here at the bottom, here's the speed. If you have a myelinated motor axon, which is what the PNS neurons are, 120 meters per second. That's really fast. A meter is about 3 feet. So it's going 120 times 3 feet. So 360 feet per second. That helps you out. That's more than a football field in length. Okay. Per second. But an unmyelinated one is only 0.5 meters per second. Much, much slower. The unmyelinated neurons are usually found in the CNS, which is your brain and spinal cord. They don't have as much of a distance to go, so it doesn't matter as much. But speed matters for the PNS. Okay. Okay. So this will be the last slide for today. I'll get into the synaptic transmission later. So there is an all or nothing response here. So either the threshold charge of negative 55 millivolts is met or it is not. So it's kind of like flipping on a light switch. The light is on or the light is off. Same thing. There is an impulse or there is not. all action potentials are of the same strength for each type of neuron. But if there's more, if the stimulation is more intense, then that triggers more impulses, but not a stronger impulse. So basically the ions that split back and forth only have this one charge and only have this one speed. But what can change is how many times you send the signal. Okay. Now, there is something called a refractory period where that specific spot on the neuron has to rest, and that's when the sodium and potassium pump is having to reset the neuron to its resting potential. So if the threshold stimulus is happening super rapidly, there comes a point where like it physically cannot cause another action potential because the neuron hasn't reset yet. So there is an upper limit on how many active potentials you can send. You can only think so fast. And the reason for this refractory period is that direction, okay, that the impulse only goes one direction. This does take a lot of energy to put the sodium and potassium back. So the more intensively you're concentrating, the more tired you're going to be overall, like from an organismal level. I read somewhere once that like a grandmaster at chess is actually burning as many calories as like a marathon runner. You know, when you take it like per minute, because they're thinking so deeply. Okay. So in the next lesson, we'll pick up with synaptic transmission, how those neurotransmitters do what they do. But we'll stop there for today.